All right, well, welcome everyone to the Clinical Chemistry Training Program information session. We're really glad you signed up and are here today. I'm going to make some few introductory remarks and then go through what our agenda is. So I first want to make some introductions to some of our panelists who are here with us today. So I'll introduce myself. I'm Vathany. I am the division head for clinical biochemistry at University Health Network, and I am one of the program co-directors for our fellowship program. I have with me two of our faculty members, Dr. Felix Lung, who is the division head of clinical biochemistry at Mount Sinai Hospital, and Dr. Lucia Safiashvili, who is our clinical biochemist at the Hospital for Sick Children. We also have today our first and second year fellows with us. So Dr. Xianyi Liu and Dr. Mash Ashwari Warihini, who is our second year.
Take off where Bethany left off. So just to introduce everyone to our administrative side as well. Ms. Paula Nixon is not here, but she actually coordinates all of the postgraduate programs within our Department of Laboratory Medicine and Pathobiology. So of which this fellowship program is one of many other programs within the postgraduate part of our department at U of T. And then finally, we have Tara here as well. Tara focuses on helping us in our program administration. I believe you have a few other programs, Tara, but we're very lucky to have Tara help us out with the administrative side of our clinical chemistry fellowship. Okay, so this will be the agenda. So we'll give an overview of the fellowship and what that program looks like across the 2 years. We actually have 2 videos for everyone here as well, and they're going to be virtual tours of 2 of the facilities that you would rotate through. We will have a panel discussion with myself and the other guests that we have here. And then finally, we'll open it up for Q&A from all of you. And I believe some of you had already submitted some questions as well. So we will try to address those. Okay, I'm really going to wing it here. So here is an example of a few things that we do in the clinical biochemistry lab. Now, many of you might not even really know what it is. High level, clinical biochemistry represents one of the major disciplines within the specialty of laboratory medicine. And when we refer to laboratory medicine, we are talking about any type of lab work that you would need to do as part of management of a patient, whether they are inside the hospital or they are not in the hospital, but going to, let's say, a family medicine clinic. So here we show, for example, what we would potentially do to assess whether or not someone has drugs in their system. So here you see 3 compounds. 3 of these probably should be quite familiar. You probably have heard of them in the news at least, but morphine, oxycodone, fentanyl. These are 3 very common compounds that we would test for in, let's say, a patient's urine, if that is what we're trying to figure out. On the right, you see an example of a mass spec or a mass spectrometer, and that is typically one of the instruments that we would use in the clinical lab in order to judge whether or not, let's say, these drug or compounds are in a patient's urine. And obviously, that result leads to some sort of management or decision down the line for the clinician who is managing that patient. Really briefly, I just want to highlight also the COVID-19 test. So I know many of you probably dealt with it as just at-home kits, but recognizing that that in itself is still representative of laboratory medicine. It's a very unique part of laboratory medicine because actually you're doing the testing outside of the lab. And that would be an example of what we would call point of care testing, right? Represents lab testing. You get some sort of decision based on it or some result that probably is going to determine what you do or what the clinician does, but it happens outside the lab.
So to give you a brief overview of our University of Toronto Fellowship Program, this was established in 1968, and we've had over 130 graduates so far. We provide a very comprehensive training in clinical chemistry with additional exposure in the other lab disciplines, including hematology, microbiology, and molecular diagnostics. This is a pathway to professional certification and practice, and our fellowship program is accredited by the Canadian Academy of Clinical Biochemistry, or CACB, as well as the COMAC, which is the equivalent organization in the United States. Our fellowship program is fully funded by a combination of both the Ontario Ministry of Health as well as the University of Toronto postdoctoral postgraduate medical education. We have over 30 plus.
Sorry, I don't understand.
Teaching faculty. So the eligibility requirements in order to be able to apply to the fellowship program, and this is all stated on our website as well, is that you must be a Canadian citizen or a permanent resident at the time of application. And you need to show proof of that. You need to have completed a PhD in biochemistry or a related science. And if you're currently in your final year of your PhD program, your PhD defense must be completed before the start of our fellowship, which is July 1st. Some of the requirements or skills that we look for is to have good communication skills, that's both written and verbal, and a minimum of at least a B+ in some of the relevant courses and an overall average during the graduate studies. So our application process, you could click on this, take your phone out, scan, to be able to see. The deadline for applications is January 15th of 2025. The applications were open on October 1st, and early interviews will occur in early February for selected applicants only, with the program to start in July. So some of the documents that we ask for during the application is a cover letter outlining your interest in the fellowship program, and why you're interested, tell us about yourself, your CV, as well as your official transcripts from undergrad, graduate studies, and if your PhD has not been awarded, we do need a letter from your primary supervisor indicating that you will receive your degree by June 30th of 2025. We will need copies of the degrees that's awarded. And for international degrees, they need to be accompanied by an assessment. We need 3 letters of references along with referee evaluation forms. When you start in the fellowship program, we have a number of hospitals that participate in the core rotations. And this includes Mount Sinai Hospital, the University Health Network, the Hospital for Sick Children, Sunnybrook Health Sciences Center, and St. Michael's Unity Health. These 5 organizations or institutions are where you will spend the bulk of your 2 years of fellowship here. During the training program, we have a number of ways to assess the candidate, and we provide both coursework as well as other activities and evaluations. When it comes to coursework, there is an analytical course on biochemistry and the different principles and technologies. So this ensures that all of our fellows, right from the beginning, are on the same playing field. So we provide an analytical course for them to take. They also participate in the University of Toronto's medical school with the first year medical students by taking some of their courses in the first year. So this gives them the clinical perspective that's necessary to be successful. And then in second year, our fellows take a biostatistics and programming course, which are essential skill sets to have, as well as a laboratory management course. And besides traditional coursework, there's ongoing activities that the fellows participate in. And some of you may have joined in on the journal clubs that we have on Tuesdays. So this is an opportunity for our fellows to select a topic and to present on it. We also have tutorial sessions. Our fellows participate in a number of projects. They attend courses and have various meetings and check-ins with the site supervisors or the teaching faculty or the program directors. In terms of evaluations of our fellows, there's a number of different ways that we assess through their performance in the mandatory rotations, their performance in the courses. There we have committee meetings where we assess their knowledge up to the point where they are right now. We evaluate their journal clubs and have what we call mock oral examinations or test of knowledge questions to them throughout the 2 years. So most of the mandatory rotations across those 5 hospitals that I mentioned probably make up approximately a year and a half of the fellowship program. And then the remaining half a year, our fellows get to select elective rotations where they can go to other academic hospitals not covered in the mandatory rotations or other hospitals within Ontario or the rest of the province. Some examples of elective rotations that our previous fellows have done are shown here. And this was a photo that we took of the faculty and graduates of the fellowship program in 2019 when we were celebrating 50 years of the University of Toronto training program. So as you can see, we have quite a diverse array of faculty and graduates from the program. So that was the program overview. We will now share 2 videos of the Mount Sinai Hospital rotation, which will be the first rotation that fellows take when they join the program.
Your first 6-month rotation in the U of T postdoctoral clinical chemistry program from July to December of your first year. As you enter the lab, you will see specimen management, where they receive and accession samples from both outpatient clinics and inpatient wards at Mount Sinai Hospital and external centers. Right next to specimen management is the blood gas and urinalysis bench. Here, technologists perform testing for analytes that are critically needed to inform care, including pH, blood gases, lactate, and electrolytes. Mount Sinai's main analyzers are from the Roche Cobas line. These core analyzers offer both immunoassay and chemistry testing, spanning most routine analytes available in the clinical lab. They are set up on a track system for full automation, including sample sorting, analysis, and storage. Our laboratory also has a point-of-care team who oversees point-of-care testing, including glucometers, at Mount Sinai Hospital, as well as several external sites. Mount Sinai's clinical laboratory also includes a special chemistry division with several analyzers, such as the Helena Touch, which is used for serum protein electrophoresis, the BioFlash, which is used to measure autoantibodies that are helpful in the diagnosis of autoimmune disease, and the Beckman Access, which is used to measure fertility hormones such as VMH, as well as analytes that are part of maternal serum screening. This program is unique to Sinai in Toronto. The special chemistry section also includes a diazorin liaison for testing of 25-hydroxyvitamin D and 1,25-hydroxyvitamin D, as well as a standalone instrument for intraoperative PTH testing to guide surgery in patients with primary hyperparathyroidism. Lastly, this is the Fellows Workstation. You or your co-fellow can use this space to work on projects, cases, and study throughout your first rotation. We hope you enjoyed this virtual tour and hope to see you soon.
So the next tour that you will hear about is the University Health Network, which is where the first-year fellows spend the latter half of the first year.
Your second 6-month rotation will be across the street at Toronto General Hospital, which is part of University Health Network. Similar to Mount Sinai, UHN also has a at Mount Sinai Hospital. These analyzers offer both chemistry and immunoassays, covering most tests available, and consist of routine test menus. These analyzers are set up on a track system for full automation. This is one of the Abbott Lineage I analyzers, where serology testing is being performed on these analyzers to detect exposure to certain viral diseases such as HIV or hepatitis B and C. These are the GEM 5000 blood gas analyzers offered at UHS, similar to those at Mount Sinai Hospital. In addition to pH and blood gas analyzers can also measure tests such as hemoglobin and electrolytes as an example. This is the Siemens Clinitec Novus urinalysis instrument, similar to that offered at Mount Sinai Hospital, which measures urinary substances to assess kidney function. This immunoassay instrument is BioPlex, which uses Luminex speed technology to measure autoantibodies for autoimmune diseases such as lupus. This is the OptiLight instrument, which uses turbidimetric immunoassay technology to measure specialized analytes such as serum free light chains workup of multiple myeloma. This is the serum protein electrophoresis instrument at University Health Network, which you had also previously seen at Mount Sinai Hospital. And so this instrument separates proteins based on their electrophoretic mobility in a gel. This is the Cryptor, which is a time-resolved fluorescence analyzer that offers specialized menu of testing, such as the chromogranin A, which is used in the workup of neuroendocrine tumors. This is a high-resolution liquid chromatography mass spectrometer, which is used to identify proteins on biopsy samples, and it's used to help identify proteins within subtypes of amyloidosis. Amyloidosis is a disease of abnormal protein folding deposition in end organs, such as kidney and heart, for example. This is a liquid chromatography triple quadruple mass spectrometer, which is currently used to measure small biological molecules such as methylmalonic acid in the assessment of vitamin B12 deficiency. Lastly, this is one of the workstations available for fellows during the rotation at UHS. As you will start your on-call duties at this hospital, you will have the workstation to review patient charts and speak with healthcare providers.
So now that we're done with the virtual tours, I just wanted to bring back our panelist members. We do have a list of questions that you have asked when you were registering, so we can definitely go through that. But if you go to the next slide, Felix. So this is just a reminder to stay in touch with us and to join our mailing list if you're interested in getting updates, application reminders, or invitations to events like this that's relevant to our postdoctoral diploma program. Please scan here and sign up to be on the mailing list. As we now open it up, we have plenty of time for questions. You can use the chat function to ask your question. You could raise your hand and wait to be called. And of course, if you have any other questions, you can definitely email me later with it.